Every evening, in small villages across western India, children run home as the sun drops and the evening smoke drifts through the doorways. But this season, many of them have a fever. That fever will climb. A headache. Vomiting. Their eyes will glaze over. And before the next two sunrises, that child may be gone. Not in weeks. In as little as 48 hours. It's not dengue. It's not the flu. It has no cure. It has no vaccine. And right now, in India, it is taking the lives of children. This is the story of the Chandipura virus, and the tiny insect that may be carrying death on its back. So let's meet the enemy. Chandipura virus CHPV. Under a microscope it doesn't look like a ball or a crown. It looks like a bullet, a rod with a rounded tip. That shape reveals its family, because it belongs to the Rhabdoviridae group, the same family as one of history's most feared diseases, rabies. Inside that bullet is a single strand of RNA, its entire genetic instruction manual wrapped in a protein shell. Sticking out of the surface are spiky protein keys called glycoproteins. Remember that word because those spikes are its tools for breaking in. It was discovered almost by accident in 1965. Scientists at the National Institute of Virology in Pune were studying patients with a fever looking for dengue and chikungunya and instead found something new in a village called Chandipura in Maharashtra. That's how it got its name. For decades it stayed obscure. Then, in 2003, it exploded. In 2003, Chandipura killed more than 300 children. It was a brutal wake-up call. But then the cases faded, only a few sporadic ones after 2010. The virus seemed to vanish into nature, waiting. And then, this monsoon season, it returned, striking Gujarat and Rajasthan, and it started killing children within days. This is the terrifying part. The virus works fast. A child may seem fine, a bit tired, a headache, body aches like a cold. But within hours it can turn serious. The fever spikes. Vomiting. Seizures. Convulsions. The brain itself becomes inflamed. Doctors call this encephalitis. In many cases, death comes within 24 to 48 hours of the first serious symptoms. Not a slow decline, a sprint, and the virus usually wins. To understand why it's so fast, follow where it goes. When the virus enters the body, it makes a direct line for the central nervous system, the brain and spinal cord. Your brain is guarded by a brilliant wall of tightly packed cells called the blood-brain barrier, the doorman who decides who gets in. Chandipura has learned how to get past him. Once inside, it multiplies rapidly. But here's the twist most people don't know. The worst damage isn't caused by the virus alone. It's caused by our own immune system. When the body detects the invasion, it floods the area with signaling proteins called cytokines, a massive inflammatory response meant to fight the enemy. But that battle destroys healthy nerve cells too. Doctors call this immunopathology. The immune response itself becomes part of the damage. Think of it like a fire alarm that keeps screaming and floods the whole building with water. The water, the inflammation, ends up wrecking the building, even as it tries to put out the fire. That's what's happening inside the brain. That's why a simple fever can become a life-threatening emergency in a single day. So where does this killer come from? Not from other humans. There's no evidence it spreads person to person. Instead, it's carried by a tiny, easy-to-miss insect, the sandfly. Sandflies are smaller than mosquitoes. You might not notice the bite. The main suspect is a species called Phlebotomus puptaceae. The virus lives in the fly's salivary glands, and when the fly bites to feed on blood, it injects the virus with its saliva. One bite. That's all it takes. These flies love damp, dark, protected spots, and especially the monsoon season, when the air turns humid. And here's a surprising everyday detail. In rural India, families traditionally use cow dung to plaster walls and make fuel cakes. That organic material is a perfect breeding spot for sandflies. So the very habits of daily rural life, keeping livestock close, using cow dung, can accidentally create the perfect home and buffet for the vector. But here's a genuinely surprising twist from this outbreak, and it's a great example of science being honest. In the previous outbreak, scientists collected and tested large numbers of sandflies, and every sample came back negative for the virus. No vector could be conclusively identified. So this year the investigation has widened. Mosquitoes, ticks, and mites are now being examined as possible carriers. Thousands of vector samples are in the lab. Officials have been careful to say it's too early to name the exact vector for the current outbreak. Now let's look at what's actually happening right now, because the latest numbers are serious. This monsoon, Gujarat has reported 184 suspected cases of the virus. Of those, 35 have been laboratory confirmed positive. The results of 11 more samples are awaited. And tragically, 22 children have died. Let me put that in plain terms. 184 suspected. 35 confirmed. 22 children dead. And as of this week, 7 children are still under treatment. This is serious enough that the government has stepped in at the highest level. India's Ministry of Health and Family Welfare has deployed a National Joint Outbreak Response Team (NJOT) to both Gujarat and Rajasthan to strengthen the response. The NCDC has also activated its Public Health Emergency Operations Center to back them up. And this isn't just a local response, it's one of the most comprehensive scientific investigations into Chandipura virus in years. A whole network of India's top institutions is involved: the National Centre for Disease Control, the ICMR, and the Department of Animal Husbandry. Epidemiologists, virologists, entomologists, and veterinary scientists are all working together. They are doing community serosurveys, testing people to find mild and even asymptomatic infections to estimate the true scale. And they are checking animals too. Around 70 blood samples from cattle, buffaloes, and goats have been collected, along with milk samples, to see if animals are hidden reservoirs. And they are doing whole genome sequencing of the current virus at the Gujarat Biotechnology Research Center. Early results show only minor genetic variations compared to earlier strains, but scientists say detailed studies are needed to know if this virus has changed to become more dangerous. Here's how the virus is confirmed. Most positive cases are found by detecting the virus's genetic material in blood or spinal fluid using a test called PCR. Others are caught by detecting antibodies called IgM. And one detail stands out across every report: this disease overwhelmingly targets children. It's the young who are dying. But let's stay honest, because a trustworthy video has to be. Only 35 of 184 suspected cases are confirmed. That gap reflects how fast this virus kills. Patients may not live long enough to produce detectable antibodies, and how hard it is to detect. So part of this story is that we still don't have the full picture, and the response is racing to catch up. This is the chapter that separates a real documentary from a sensational one, admitting what we don't know. For Chandipura, that list is long. First, the animal reservoir. We are testing cattle, buffaloes, and goats right now. In the past, animals in endemic areas have shown antibodies against the virus, but some of those samples also reacted to rabies, so scientists couldn't be sure. Is there a hidden animal keeping this virus alive between outbreaks? We don't know yet. Second, the virus's genetics. Virus samples from 1965 to 2007 were more than 97% identical, barely changed in 40 years. Yet the 2003 strain seemed more virulent, more deadly than the original. Why some versions kill more than others is still a mystery. And genome sequencing of this year's virus is only just beginning. Third and deepest, the exact mechanism of how the virus crosses the blood-brain barrier and invades the brain while dodging our immune defenses. Its replication, its interaction with our cells, the triggers of nerve cell death, almost all of it remains unknown. So the hard question, what can be done? Let's be brutally honest. Right now, very little. There is no approved antiviral for Chandipura. And there is no commercial vaccine. Management is purely symptomatic. Doctors control brain inflammation and support the patient. But because the disease races so fast, that reactive approach can run out of time. In the lab, two existing drugs, favipiravir and ribavirin, show antiviral activity against the virus in test tube studies. But their effectiveness in real patients isn't proven. A glimmer of hope. Not a cure. Vaccines are where the story gets interesting and frustrating. The National Institute of Virology has developed two vaccine candidates, one inactivated, one recombinant protein subunit. In mice, both triggered strong immune responses and provided complete protection against a lethal dose. Read that again, an effective vaccine, working in the lab, fully protecting mice. So if there's no vaccine and no cure, what's the most powerful weapon we have right now? Prevention. Stopping the bite. This is where the fight is actually won or lost. Reducing breeding sites, clearing damp, dark spots around homes. Improving sanitation. Being thoughtful about livestock and cow dung storage wherever practical. Insecticide spraying in high-risk areas, especially during monsoon. And public awareness, helping families recognize early symptoms and get help fast, because time is the scarcest resource here. But prevention can't be left to families alone. It needs policy, governments investing in it needs research, faster diagnostic tools that can confirm Chandipura in the field where a big referral lab isn't close by. And behind all of it is a modern idea called One Health, treating human, animal, and environmental health as one connected system, because a disease that lives in insects and animals can't be solved by looking at people alone. That's the smart way to fight these outbreaks. Now, why should you care? Because a virus that hides in an insect, kills in 48 hours, and has no cure is a window into something bigger. It shows how quickly young lives can be lost to something preventable if we don't invest in science, surveillance, and prevention before an outbreak, not after. Let's bring it all together. Chandipura virus is a rare but devastating cause of acute encephalitis in children in India. It's transmitted by insect bites, most likely sandflies, not person to person. It moves from a flu-like illness to brain inflammation to death in as little as 24 to 48 hours. This monsoon, Gujarat has reported 184 suspected cases, 35 confirmed, report Science. Look at organizations and researchers working on vaccines for neglected diseases. Third, stay curious. Nature is already planning its next experiment. The question is whether we learn the lesson this time, or wait for another outbreak to remind us. If this helped you understand something new, hit like, subscribe, and turn on notifications, so you can think clearly when it matters. Thanks for watching. Knowledge is the best vaccine we have. See you in the next one.